immunology, antibody, structure and function, and immunoglobulin isotypes. So first off, let's talk about the antibody. We know that the antibody is going to be produced by the B cells first and foremost, okay? And what they want you to know is that it's broken down into multiple structures, okay? So take a look at what the antibody looks like from a diagram perspective. It's very easy to understand. So first, you have a constant region and a variable region, okay? The variable region is going to essentially change in somatic hypermutation, okay? And so this means that in order to change the ability to bind different antigens, you have to undergo somatic hypermutation, which changes the affinity in which you can bind different pathogens or antigens. And so that's going to be the variable region changing. The constant region will change during isotope switching, meaning when you're changing from a certain immunoglobulin to another one. So from IgM to IgG or IgM to IgA. That's when you will have the constant region changing. Okay, so remember those two types of things. Somatic hypermutation and affinity maturation is going to be due to variable region changing. Okay, so if you want to change essentially what antigen you bind. And then the isotope switching is going to be due to the constant region changing, which is when you want to change what immunoglobulin you are from IgM to IgG. All right, so that's the first step. The second step is to kind of recognize the second portions of the regions. You have the Fab fragment, okay, and you have the FC fragment. So you have the FAB fragment and the FC fragment. So the FC region of the fragment, usually on IgM and IgG, it fixes complements and it attaches essentially to our immune cells. Okay, so this is where it will bind to our immune cells to help bring in the pathogen. The FAB region of the fragment is going to be the one in which it binds and recognizes the antigen. Okay, so those are the two differences between FAB and FC region. All right. Lastly, there's the hinge region. If you increase the hinge region, the more hinge you have, the more avidity you have. Okay, so IgM, for example, has the largest amount of avidity because it has the most amount of hinge sites. Okay, so keep in mind all of the structure in which the antibody can change and function. Remember, somatic hypermutation and affinity maturation is going to change the variable region. Okay, whereas if you want to change the isotype, for isotype switching, it's going to change the constant region. Okay, moving forward to immunoglobulin isotypes. So these are our specific types of immunoglobulins. So mature B cells, okay, are going to produce IgM first. Okay, so they're always going to produce IgM first. And then they can do class switching. All right, they're going to produce class switching. So let's see the different types of immunoglobulin isotypes that it could class switch to from IgM. So IgG, all right, IgG is the main antibody in secondary response. So it's the most abundant antibody. It can cross the placenta, is very key. Okay, very, very key. It's associated with autoimmune hemolytic anemia, specifically the warm type. All right, that's an associated pathology. Next is going to be IgA. IgA is going to be for our mucous membranes, our mucous membrane site. So it's in the GI tract at the Peyer's patches. It's in breast milk. Okay, it's in our secretions of tears, saliva, and mucus. Okay. It's going to protect all of those mucous membranes. Next is IgM. IgM is the first antibody to be produced, as we said, and it has the highest avidity. Avidity is essentially the ability to bind antigens because it has 5 antibodies all connected. It's a pentamer. This is associated with cold autoimmune hemolytic anemia, that type of pathology. All right. IgE is the last important one. It binds mast cells and basophils. And what it does is it cross-links them when exposed to an allergen. That cross-linking is going to cause an immediate type 1 hypersensitivity reaction, causing a release of histamine. All right. This also, IgE contributes to protection and immunity against parasites by activating eosinophils. All right. So that's kind of our end-all be-all for our immunoglobulin isotypes. Don't forget the structure and the certain types and what they're associated with.